Valentine's Day to everyone, and what better way to celebrate Valentine's Day than to welcome back a very special past a colleague that was of ours, Tanya White. Really excited to introduce Tanya today. Tanya, as many of you know, was a faculty here in the Department of Psychiatry for 9 years or 8 years in the in the early part of the century. So a little bit of background about Tanya. She started out her education in electrical engineering. She had a bachelor's and a master's in electrical engineering, but then she went to medical school at the University of Illinois, and then she started her research career doing a postdoc, learning neuroimaging, brain development, and schizophrenia, and worked with Nancy Andreasen until she was recruited by our past chair, Dr. Chuck Schultz, to come and work with us here in the department in a tenure-track position back in 2001, or 2001. And she really was a wonderful faculty member with us. She started a youth psychosis program at that time, which is still thriving, and she got a K award and studied brain imaging and early psychosis, and established a lot of collaborations across the university in child development and neuroimaging and other fields. And she was a mentor to me and many others. She really was my first research mentor, and really taught me what it's like to be a successful researcher, and what is that like to be a clinician and a researcher at the same time, and how to persist after getting rejected from things and things like that. Tanya was recruited away from us in 2009. She has been at Erasmus University in the Netherlands studying brain development, taking the lead on the neuroimaging component of a really large, exciting study there called the Generation R study. So she'll be sharing some of that with us today. Please, so I guess I'll wrap it up there. Please join me in welcoming Tanya White.
Thanks for the lovely introduction, and it's wonderful to be back and after 8.5 years and to be able to give a talk to let you know what I've been doing since I moved to across the pond. And when I moved, one of the things that was really intriguing about the Rotterdam, or about the study going on in Rotterdam, which is this intersection between epidemiology and neuroscience. So when I was at Minnesota, I had a youth psychosis clinic, as Katie mentioned, and what they were doing, or what they needed as part of the Generation R study, which is a large study of child development, was to infuse neuroimaging into this program, which, and I'll give you an overview of the Generation R study, and as I talk about it, you'll see the opportunities of this study to look at psychopathology before even the prodrome. So emerging psychopathology. So I gave a talk yesterday at the Center for Neurobehavioral Development, which was wonderful. And there I focused in on more specific areas, and I decided today to actually do more of a smorgasbord approach, to not go in-depth in topics, but to go superficially over the different domains of studies that I'm involved with in the Rotterdam. So first of all, the Generation R study falls into this intersection between epidemiology and developmental neuroscience, and we also have some studies looking at imaging genetics, which I'll also discuss. And epidemiology, in terms of large pediatric neuroimaging cohorts, I divide them up into different categories. So there's some that are typical development, which I differentiate from those that are population-based. So you can see the population-based studies on the right, of which Generation R is one of those. The difference in that is that really they try and recruit as much as possible from the general population without much in the way of exclusion criteria. So try and be overly inclusive to be representative of the general population. So the generation R study is and started as an epidemiological study in the true sense. So within Rotterdam, I don't know if I have a pointer. It's okay. So this is Rotterdam in the Netherlands, and the inclusion criteria was only two things. Mothers had to be expecting between 2002 and 2006, so they were pregnant and expecting, and they lived within certain zip codes in Rotterdam. And that was the only two, thank you. That was the only two inclusion criteria. The plan is to follow the children into young adulthood, and the children are now 13 to 14 years of age. And this is not just mental health. This is population-based study covering multiple domains in pediatrics, ophthalmology, hematology, immunology, dental. So it's a multifaceted study. And I'll get into that more. Rotterdam is the largest port city in Europe, and it's very ethnically diverse, with 58% Dutch and other Europeans. But then you can see that the ethnic percentage of the group is quite diverse in ethnicities, which sometimes when we write NIH grants, it's actually hard to put these individuals into different categories, but these match well with the population within Rotterdam. So the Generation R study closely resembles the population of Rotterdam. It doesn't necessarily represent the population of other cities in the Netherlands because of the Rotterdam tends to draw large ethnic populations. So I'm going to run through really briefly some of the measures that we've collected within the Generation R study. The first study, the first metric that we have of brain development is actually in utero with brain ultrasound. And I'm going to show you some of the results of our findings with brain ultrasound as I go along. So we have three different measures of ultrasound of the head in the first, second, and third trimester. And then in a subgroup at 6 weeks, we had a postnatal ultrasound through the anterior fontanelle. Blood has been collected at multiple time points in the study. There have been multiple questionnaires. The families, if you tabulate all the different variables that have been collected, I think it would probably reach the moon. There's just tons of variables. The family should get an award for all the questionnaires that they filled out. But they go back to prenatal life, including demographic characteristics, lifestyle, diet, smoking, psychosocial characteristics, as well as postnatal, being child temperament, behavior, sleeping, sports, television, cognitive measures, and parental psychosocial characteristics. One of the main behavioral metrics that we're using is the child behavior checklist, which has been collected repeatedly. We're currently on our fifth measurement wave of the CBCL, so we have longitudinal data looking at internalizing, externalizing, and then the specific domains in the CBCL, as well as cognitive domains. The brief was collected at 4 years of age. We have other measures in subgroups, inhibition, working memory. We had the entire cohort did a nonverbal IQ at the age of 6. We have collected IQ in the mother, and we had a subgroup who performed a neuropsychological test using the NEPC-2, and we're currently doing the WISC-5 on the entire sample. We've had behavioral and psychiatric interviews occurring at the time of birth, but also we had at 6 years of age the Berkeley puppet interview in about between 5,000 and 6,000, and we've also had the CDISC, the child diagnostic interview for diagnostic interview schedule for children performed between 6 to 8 years of age in a subgroup that were high risk for psychopathology. So I want to show you, so imagine this is a preview of the Berkeley puppet interview, and this was collected in about 5,500 children in almost an assembly line fashion in the Generation R Research Center.
Hey, en hoe heet jij? Neliana. Hoi Neliana. Hey Neliana, hoe gaat het met jou? Goed. Nou ja, met mij ook. En met mij ook. En ik heb niet veel enge dromen. En ik heb wel veel enge dromen. En jij? Niet. Ik ook niet veel. En ik heb veel vriendjes en vriendinnetjes op school. En ik heb niet veel vriendjes en vriendinnetjes op school. En jij, Merel? Wel. Hey Iris, als kinderen spelletjes spelen, dan mag ik niet meedoen. En als kinderen spelletjes spelen, dan mag ik wel meedoen. Soms dat ik niet mee mag doen. En soms dat ik wel mee mag doen. Hey, en weet je Zep, als ik op school ben, dan mis ik mijn mama of papa. En als ik op school ben, dan mis ik mijn mama of papa niet. En jij? Ik niet, omdat ze komen nog terug. Ja, precies. Dat is net als ik. Ik doe niet veel. Ik huil wel veel. En jij? Nou, ik heb niet echt veel. Nee, ik eigenlijk ook niet echt veel. En soms dan sla ik mijn juf of andere grote mensen. En ik sla mijn juf of andere grote mensen niet. Dat doe ik nooit, omdat mijn juf is hartstikke lief. Oh, weet je, wij vonden het wel heel gezellig om met jou te kletsen. Hé, maar dan gaan wij nu echt weer hoor. Nog veel plezier hè, vandaag. Hey, dag Demi. Dag. Dag.
So that's one of the measures that was collected at the research center called when the children were 6. It's the Berkeley puppet interview. So one of the challenges is that since there's multiple groups that are involved in the study, we have a certain amount of time, so our time is limited because they go from station to station, and they've got one station in where all the behavioral metrics are collected, and then they have a DEXA scan, they have pictures for their dental work, they have ophthalmology, they do pulmonary function tests, they have a cardiac echo, and so they go from place to place and measures of fat, looking at obesity. So it covers multiple different domains, and then we have body materials that are collected. So, and the imaging, which I'm going to get to, is a completely different visit. The other thing that we've done in a subgroup is looking at measures of bullying. So one of the PhD students on a grant went into the schools and did a peer nomination with all schools within the Rotterdam area. So since this population-based study takes place within Rotterdam, if we do it in all children, we know which children are also a part of the Generation R study. And we can look at victimization and bullying behavior in those children. And then I mentioned the diagnostic interview schedule for children. That was completed in a subgroup as well. MRI. So I moved in 2009 to, there was no MRI imaging at the time, so my job was to infuse neuroimaging into the Generation R study. And it's a team effort because we did large amounts of neuroimaging data collection, and we're currently on our third wave. And so we started in September 2009, and we had, by July 2013, 1,070 6 to 8-year-old children scanned. Sometimes this feels like McDonald's, how many hamburgers served. And in 2013, we had a dedicated scanner installed, and we began Wave 2, and we've scanned 4,150 9 to 10-year-old children on one scanner. So this is on one scanner, averaging 130 to 170 children per month. And we began scanning Wave 3 in October 2016, and we've reached over 1,500. Because the Netherlands has really quite good healthcare, we're having the challenge that about 30 to 40% of the children have braces. And that doesn't bode well for certain sequences. So we came up with a structural image sequence that we can use for braces, but it's still a challenge. So we do a structural MRI scan, diffusion tensor imaging, which is 35 directions, and a resting state functional MRI as part of our data collection. We have this for 30 minutes because there's also a 30-minute body scan that's done looking at heart, lung. They do actually pulmonary function tests in the scanner. They have a body scan looking at body fat and liver, kidneys, and then hip scan and gonads to look at pubertal development. So image quality is something that's often a challenge when scanning children. And one of our techs started showing the children an image when you lay still versus if you move a lot, and actually this visual representation helps them, they visualize what happens when they move, and it actually helped the quality of the scan, which I will go into a bit. So we had difficulties with the Medical Ethics Committee, the IRB. In the Netherlands, it's a Medical Ethics Committee. So we wanted to start at 6 years. They said we needed to start at 8 years, but we were able to move it down to 6 years. And part of this is similar to what Sarah Durston uses in Utrecht. And we have smiley faces. That's an intermittent. We have smiley faces here. So we asked the children before the mock scan, after the mock scan, and after the MRI scan about their experience. If they were afraid or if they enjoyed it, we asked them 2 different questions. And if they ever point to a 5, we stop. We stop the scan right away. So this allows the children to give informed assent for their participation, and they know that. But you can see most children do really quite well. And in fact, here's how the children, level of anxiety, here's how the children rate themselves versus how the parent and the researcher rate the child's level of anxiety. So the children rate themselves less than what the researchers and the parents rate, what they think the child's anxiety is. So when you have so many scans, some of these things we need to automate. So I'm going to go really briefly through an auto quality for structural images that we developed. And here's a really good quality image here. And you can see the waves, the ringing and the waves here in the poor quality image. But you can see there's really nice borders with the outside of the brain. But if you just give this a different threshold, you can see, because of the way images are collected, that these are the same images just with a different threshold. You can see outside the head, there you can see waves, where there's much less here. So this is a bit thinking outside the box or thinking outside the head here. Can we measure this, the gradient? We measured several different things here, but the gradient of this edge to look at image quality of the scans through a specific algorithm where we look at edge detection, and then we quantify this noise characteristics outside the brain in this area using several different approaches, and actually the gradient worked the best compared to the Fourier approach. So we have, of our scans, if image quality is poor, then we repeat the scan. So we had, out of our first 3,053, we had 2,758 scans that were not repeated, and 294 scans were repeated. So one of the questions we ask, can we predict which scans are repeated? And that, here's our 3 different approaches. I said the area under the curve here in terms of predicting which scans will be bad, and the gradient approach did very nicely in predicting area under the curve. In the Generation R study, we replicated this in the first and second wave of the Generation R in the NIMH data from Philip Shaw and in the Gusto study in Singapore, and found very similar results. And then we asked the question, if they have a poor scan, if the children have a poor scan, is repeating the scan, does it help? And here's all the scans, the quality. And yes, if you repeat the scans, the second scan is actually better. So there's probably some adjustment to the scanner environment, and we do get better quality in repeating the scans. Finally, and perhaps most interesting, is that this is a relationship, a correlation between the auto quality assessment rating in the child and a certain measure of cortical thickness in different lobes. So, and what we're doing is removing the worst quality here. So they're sorted by worst quality, and each of them are removed. And actually, what you see here, what you'd like to see is there be a certain threshold here where you exclude a number of subjects, and you know that there's no relationship between this measure, which is actually outside of the head, outside of the brain, and your measure inside of the brain. But it doesn't happen. It's almost a linear relationship. So the thought is that this metric, that quality of the images, has a subtle effect that we should look at and control for in our imaging studies. Now, from here, I'm going to walk you through give you a smorgasbord. We're in Minnesota, so smorgasbord is a word that fits here. Of 5 major research lines, and give you an example of things that are going on within the imaging group in these different areas. One of the things I'm going to start with is this is actually more in the area of epidemiology. And from an imaging perspective, this is really exciting because in many areas in imaging, it doesn't directly translate to clinical practice or some aspect. But in this area of intersection between epidemiology and developmental neuroscience, what I'm going to show you has direct public health message that is directly translatable to potential primary prevention. So I'm going to start with a question. Can good nutrition during pregnancy prevent psychiatric disorders? So there is evidence of this, right? From the Dutch hunger winter in 1944 and 45 in the Netherlands, an area that includes Rotterdam, was cut off. So the Allies were coming up through Belgium. This area within Rotterdam, Den Haag, had very little food. And so actually you can see the US and England had permission to actually drop food, and their planes weren't shot down during that period of time. But the mortality was twice the normal rate. Fertility dropped to less than half. There was an increased rate of neural tube defects, and there was a 2-fold increase in the cumulative risk of schizophrenia, as well as intracranial volume differences. So this is studies from the Dutch hunger winter. There was also a similar study showing during the Chinese famine, again, 2-fold increase in the risk of schizophrenia. Now, famine and the stress is much different than just low folate and nutritional within the general population. So, but we did ask the question with folate, because we know that folate is associated with neural tube defects, and that's very common. But the question is, low folate in the absence of neural tube defects, can that have subtle effects on brain development? So why might it have subtle effects? Well, first, we can't synthesize it, so it comes from the diet. There's an increased demand during pregnancy, and it's involved in numerous biological processes. And in the Netherlands, actually, if you look at the folate intake is in about approximately half of the population is suboptimal. And you can see here the average daily intake of folate in the Netherlands is one of the lowest. Part of that is due to early Dutch laws about not adding any anything to supplements, any supplements to food. That changed with the emergence of the EU to some extent, but this was an early study in 2002 to 2006, and so we had still many mothers with low folate. So we asked the question first, so ultrasound, we had ultrasound measures obtained at 3 points during pregnancy. We had 822 mothers with low folate in our hospital, less than 8 nanomolars per liter, and we had typical folate levels. And we were looking at growth of, ultrasound measures of head circumference. So actually we have different measures, but we're looking mainly at head circumference. We can also, in the second and third trimester, measure atrial width of the lateral ventricle and then cerebellar diameter as well. So those are measures. So the results of head growth was that head growth in children about 0.1 millimeter less per week compared to controls, which you might say 0.1 millimeters less per week is not all that big, but if you look at, take the circumference and figure out what the volume is, and then obviously not all this volume is related to neuronal, but you have, millions and billions of neurons and synapses that are probably less, and I think it's probably a good thing to have as much brain material as possible. So we were curious about, so we're doing imaging later. So the question is, what happened 6 to 9 years later? So if you would have asked me back then, I would have said, well, there's so much that occurs during early childhood and so much brain plasticity that it would be removed. It's just removed by all the other events that occurred and the plastic changes in the brain. So we looked at, we looked within a Dutch group of Dutch mothers because dietary differences are quite different when you have a smaller neuroimaging population, which was in our pilot study. That's what we had. And 62 Dutch mothers, again with the same thresholds. We used high-resolution structural MRI and found that in the same group that had decreased growth, they also had 6 to 8 years later lower volumes, lower total brain volumes. So that persisted. So actually, the goal of pregnancy is to ensure the developing fetus has optimal nutrients to assure the best chance for optimal development. So there's an important public health message. Even the absence of neural tube defects, low folic acid during pregnancy does appear to have subtle effects on neurodevelopment that are lasting. So this is common knowledge, right? This is nothing new, but it's more information to help pregnant women take folate. So what about smoking during pregnancy? So these studies started actually before I came with ultrasound data with Sabina Rosa, publishing in 2007 that this is gestational duration in weeks. So the gray line is actually the mothers who quit smoking when their pregnancy is known, and this is the reference. So if you're smoking and you learn that you're pregnant and you quit, there's no difference in head growth during pregnancy. Those who continue to smoke have a decrease in head circumference. So we also looked at this with imaging, with 113 exposed prenatally tobacco, with one-on-one matched, with controls, trying to match on all the other different characteristics. One of the things you can do in a population-based study is try to match as much as possible on all the different variables. And we again looked structural brain imaging and this time using a surface-based approach, and also found very similar results. Actually, no difference in the mothers who quit when they learned that they were pregnant, and those who did continue smoking had a pattern of rather widespread differences of decreased cortical thickness in multiple areas of the brain. We didn't find differences in volume. It was just cortical thickness for smoking. Finally, this a study on thyroid in pregnancy, and a study that is actually has changed to some extent some of the clinical practices in endocrinology related to thyroid, and because it was thought actually originally that hypothyroid is bad, hyperthyroid during pregnancy is no big deal. So we had 3,727 mothers and child pairs, of which 652 of those children with MRI, and we looked at T4 in the lowest 5% of the sample versus TSH in the normal range. And children of mothers with hypothyroxemia had 4.3 points lower IQ, and on our original study, which we were going along the lines of hypothyroidism is bad, where we didn't look at hyperthyroidism, this is what we found. But Tim Korvar in endocrinology actually looked at this bimodal approach, and here's child IQ and related to TSH during pregnancy and free T4, and found this inverted U shape related to both IQ and total gray matter volume. So psychopathology across the continuum goes along with more of the RDoC approach, in which is that let's not look at, or let's also consider psychopathology along the continuum. So in this, I'm going to move to autism spectrum disorders. So one of the other advantages of a population-based study is you have these that are clinically impaired with certain disorder, for example, autism. Oftentimes, the studies compare these with clinical autism to no problems. The children with some problems never go into clinic. They've got maybe some autistic symptoms, but they're not help-seeking. And in a population-based study, you get the full spectrum of symptoms of whatever disorder that you're looking at. And John Constantino has used the social responsiveness scale and shown that autistic symptoms do form a continuum within the population. So one of the questions that we asked was, if autistic symptoms form a continuum in the population, do the neurobiological relationships between autistic symptoms also show a continuous relationship in the population, but also related to the rates of autistic symptoms? So in this case, we took a three-prong approach. We looked at autistic symptoms across the entire spectrum. So, and with the question kind of that we all are on the autism spectrum somewhere. Some with no problems and some with some, but this is a spectrum disorder. So we then looked at what's normally done in the literature, case control design, and then we dropped children with the highest autistic symptoms. So we're going to drop the most severe clinical symptoms and say, does this relationship hold even when we exclude those with autism or high autistic symptoms? And the punchline is that it depends on where you are in the brain. So this is, we found Small differences in cortical thickness, not as large spread as what we would predicted, but we did find widespread differences in gyrification, which has also been reported by others. So when we looked at some of the different areas of the brain, so this is the right temporal frontal region, and this large area here is temporal precuneus, and you can see the fusiform gyrus here that's involved. So what we want to know is, are these areas, what kind of pattern do they fit across the continuum? So to show you that, I think this is a nice representation of two different regions. This is a left hemisphere temporal precuneus area, involving also the fusiform gyrus. And you can see this is higher autistic symptoms along here. This is gyrification index here. So as you go to higher autistic symptoms, there's almost a linear relationship with between the two, between gyrification. So this area was related even after you exclude those with high autistic symptoms. Whereas this area of the right hemisphere temporal frontal was more, if you have a large load of autistic symptoms, then you have it, but a lower. There was no relationship in some areas when you looked across, when you excluded those with high autistic symptoms. So it's probably a mix. We also did this with the NEPSI, the neuropsychological test, and actually I was hoping that it would be like more of an inverted U. Some autistic symptoms would be actually higher cognitive symptoms, but that's not what we found. We actually found, using the three different approaches that I mentioned, almost a linear relationship with worse cognitive performance globally that was related to higher autistic symptoms. So, emerging psychopathology actually is one of the areas that was very appealing when I moved to Rotterdam, because I thought we're going to have brain images of thousands of individuals potentially before they even develop psychopathology. Hopefully none of them will, but we know that some of them will develop psychopathology, depression, psychosis, and other problems. So I was looking at a grant trying to find a good model of emerging psychopathology, and there's the dysregulation disorder phenotype. And what's interesting about this, it's a behavioral phenotype in which they have elevated scores on the child behavior checklist on attention, aggression, and anxiety and depression, a prevalence of about 0.7 to 2.5% of the population, and there's been high heritability in twin studies, and it's a high-risk group of children. And if we look at the different studies in the literature, some of them longer term than others. For example, here's the number of the studies. So here's 2076. This is a South Holland study, 14 years. But what's interesting about this high-risk group is that there's high risk for psychopathology, but not one specific psychopathology. So, for example, mood disorders have a long-term risk of mood disorders, substance use disorders, personality disorders, and anxiety disorders in different studies. So it has this non-specific risk. So Marcia Buston was a PhD student who looked at using the CBCL and latent class analyses, tried to define these children across all the different CBCL scales, and came up with this group that was about 2% of the population that were really, I guess you'd call these the most children with the most behavior problems in the sample. So, whereas we also had an internalizing group and an externalizing group in this latent class. These children in the dysregulation scale were highest across essentially all domains. So we looked at nonverbal IQ in these children, and what was interesting is that they were approximately 11 points lower. Essentially, here's the internalizing, externalizing, no problem score, 11 points lower on nonverbal IQ. When you control for maternal IQ and parental psychopathology, there's still a 7-point difference. So this is a group where you say, okay, they have the most behavioral problems, and they have cognitive problems. There's something going globally in the brain. We also performed the cognitive assessment 2 years later. And then the picture actually changes a little bit, in which here's the dysregulation children. And they still are very poor, but now just poor on sensory motor, and they're poor across the board, but now there's more overlap between the internalizing and externalizing symptoms compared to the reference unit. So then we did MRI of these children. We looked at structural, diffusion tensor, and resting state MRI. So these, in theory, should be the most severely affected children in some respects. And we've had other studies showing differences with children with attention problems. I've shown you our data with autistic symptoms. But structural, we found nothing. Diffusion tensor imaging, we found nothing. And resting state fMRI, we found nothing. So a negative study on what we would have predicted to be the most problematic children. And I just received a grant to look at why that might be, because it's a bit perplexing. Is it that symptoms are less stable in childhood? Is it that there's developmental noise that is affecting these, although we have shown differences in other groups? Or is it the heterogeneity involved in the group? So we're going to look at different measures to try and parse out why we don't see things, and also just behaviorally, what happens with cognitive function and other aspects in these children that supposedly are the most severely affected. So I'm going to move on to imaging genetics, and a hot topic. It has been a hot topic, but maybe it's starting to taper off as a hot topic, as this notion of polygenic risk scores. So there's large genome-wide association studies in different disorders like schizophrenia that have highlight 114 genes, for example, that are 108 that are related to schizophrenia. And you can take these genes or the SNPs or the genome and develop a polygenic risk score for risk using those, or using a subset, actually using the entire data from the genome, or specific thresholds, because we do different amounts of selecting how significant the genes are and extracting certain groups that are related. I'll get into that as I go on, because I'm not explaining that really well. But what you do is you take an independent sample in which the GWAS is selected from. So in this case, we're using the Psychiatric Genomics Consortium, the PGC, and we're looking here at child behavior checklist measures of internalizing and externalizing scores. We're doing this in the European ancestry. So here's the total N for each of the groups, and here's the N of actually we're using the analyses. So the polygenic risk scores are performed on, calculated for schizophrenia, major depressive disorder, bipolar affective disorder, autism, ADHD, and measures of cognitive function, which is educational attainment is what we're using for that. So you're developing a polygenic risk score from an independent that was derived, the weighting is derived from an independent sample and applied to the Generation R sample is what we're doing. And here's the results. And so this is the explained variance, R squared here. This is the 3-year-old children here. This is the 6-year-old. This is the 9-year-old. The externalizing score here, internalizing score here, and here are the different disorders. So let's focus on schizophrenia. In schizophrenia at age 3, polygenic risk score for schizophrenia was related to both externalizing and internalizing scores. It remained significant at 6 years, but at 9 years it was just the measure for the internalizing score. Educational attainment was also significant at different time points, but here it's only the externalizing score was related to educational attainment. And that was negatively related. So greater externalizing scores, lower educational attainment. This was positively related. So higher polygenic risk score for schizophrenia, more internalizing problems. We didn't find anything for ADHD, for bipolar, major depressive, and autism spectrum disorder. No relationship here. The educational attainment independent sample had by far the greatest N, so it had much greater precision, and schizophrenia was second. So part of it may be the level of precision. So we were also curious about, okay, so we show a relationship between polygenic risk score for schizophrenia and for educational attainment with behavior. What about the brain? So we're looking at both structure and DTI. I'm going to show you the DTI, but actually they're both, the results were both very similar. We had 1,139 with European ancestry, and we used 35 direction diffusion tensor imaging for this study, and looked at a number of different white matter tracks within the brain. So these show you the tracks. This is actually the results, but I'll show you the results in a different metric, and that's here. So I'm going to start with schizophrenia. So this is, again, explain variance, and the stars mark the significance. We didn't find anything, any relationship between the polygenic risk score of schizophrenia and white matter. So this was negative for that, which is, we expected there to be something because we had a relationship with behavior. We did find it with ADHD, and educational attainment was also quite strong, a relationship between higher white matter microstructure and education and polygenic, I don't want to call it a risk, because it's not a risk for educational attainment, polygenic score for educational attainment. So one of the things actually we're working on now is, if ASD or other psychopathology is related to multiple genetic factors, what about multiple environmental factors? Because much of the time in epidemiology, you're looking at one specific exposure, vitamin D or fatty acids or folate or that. But what if we were to look at multiple create a poly-environmental risk score? And the Generation R study has many, many different variables, so we have the opportunity to do this. And I'm going to just give you a kind of a brief, go immediately to the results here. The thought is that if you have a poly-environmental risk score on the x-axis here, if you develop that, and a polygenic risk score on here, then you would have a gradient here of higher poly-environmental risk and high polygenic risk. You would sit with a much higher risk. So we looked within about 1,000 different variables within the Generation R study, and the red is the children with ASD, and the blue are the children without ASD, so this is more of a case control type study. You can see that you can't really pick out children with ASD, but if you have a good imagination, You can draw it here, and you can see that there's no red. So there's some signal in here, but there's certainly a lot of overlap, which is not unlike many of our studies. We find huge amounts of overlap. Now, I'm going to finish with typical development, and I already showed you some of our methodologic development. I'm going to talk about resting state networks in school-age children. And if any of you knew Ryan Metzel, who is also here from Minnesota, this is the first project I gave him when he came. He was fabulous, worked on it hard, and actually last year he received the prize for the best paper in human brain mapping. So he did a tremendous amount of work on this paper, and one of the things that we did, we looked at not only resting state networks within children, but we looked at also what are the noise components, and we looked at how stable are those networks. And so first of all, are resting state networks in school-age children similar to those in adults? So here are some typical resting state networks in adults. And actually, if you look at children, we get, as you go through the different ones, actually the visual, we get one merged network in the visual cortex, but we get the insular, temporal, we have the left parietal prefrontal, we have the right parietal prefrontal in our Generation R study. We see the default mode network, but it's divided actually into a posterior and an anterior portion, as is often seen, and it becomes merged as children get older. And we also see the frontal. So these are the most common ones that we have there. But what we did was, within our sample, we pulled out 50. We ran independent component analysis, threw them back in kind of a bootstrapping approach, pulled out another 50, threw them back in, pulled out another 50, threw them back in. And we looked at all the different components. We did that 1,000 times to see how robust these resting state networks are in school-age children. And here's the results. You can see that some of them, this is percent times that we see those resting state networks in 1,000 samples, extremely consistent. So the auditorial, the parietal default mode network, visual. Fortunately, the right and left parietal prefrontal, which are often networks that we are curious because we had another study showing that's related to IQ, and there's been others showing this PFIT, the parietal frontal integration theory, related to IQ. But then there's other ones, other networks that are much less robust. So if you find a study that finds a difference in these networks, then it could just be these networks aren't very robust, and it shows up. The gray area is noise networks. You can see some noise networks are very consistent as well, especially flow artifacts tend to be very consistent, but some movement artifacts as well are very consistent. And these white, we don't know if they're noise, we don't know, it's unclear what kind of networks they are. They kind of sit in between noise and that. But in transparency, we show all of our data and all the findings because I think it's helpful for that as opposed to showing just a subset of the resting state networks. So I'm going to summarize here. I think I'm within time, actually. So this merging field of epidemiology and developmental neuroscience is really an exciting field because it gives you not only the opportunity to look at public health aspects of research that has a direct translation, but we can look at things across the continuum where you're already recruiting subjects who may develop these subclinical symptoms very early. So you're not recruiting people with subclinical symptoms where there might be more bias in selection bias that comes later. I didn't talk about bias, but there's always bias in all our studies, and we try and control for it. So there is some attrition. So we still have about 7,000 families still involved in the studies, from close to 10,000 down to 7,000. I tell people, try and do that in the U.S. Netherlands is a small country. If people move from Rotterdam to Amsterdam, it's 30 minutes by train. Utrecht is 38 minutes by train. I made it many times when I went there for my Dutch lessons. And you can bring people back. And people, we don't pay our subjects. They just come. So it's actually very amazing and very research friendly. But there are numerous challenges. The retention of subjects is still, especially the ones who we most want to study. There's 83 children from medical records with autism spectrum disorder, but there's 54 that are still involved in the study. So it would be really nice to have all of the children involved. And there's likely going to be a dropout in those who are at increased risk for schizophrenia. Potentially. And then there's data quality issues with large samples because we have to look at tons of data. And so some of the smaller studies that do, for example, in FreeSurfer, the correction, the manual correction, we don't do that in our study. We tend to just exclude studies as opposed to doing, because we just don't have the manpower. And maybe we could do some type of citizen science to send it out there to have other people, like they do in astronomy to find new stars. We'll send something out for people to correct our neuroimages and send it back. Finally, epidemiologists have, in my experience, been less open, at least in Rotterdam, for data sharing. So I've been working on it, but moving it in that direction because we have a tremendous data sample that I think would benefit from. I tell people, if we keep it all to ourselves, then we say we have the best ideas, which we do, but no, in sharing it might would be helpful. So we just recently got a NIDA grant to parallel the ABCD study to some extent and to open up data for data sharing. This past month, I've spent a lot of time because the EU laws tend to be much more restrictive on data sharing and data protection than US laws. So I'm meeting with attorneys and talking about this and talking with people at NIDA and Greg Farber at the NIMH so I think at this point in time, it's going to be in the hands of the attorneys to try and figure out if the US will be willing to change things so that they can assure that they will meet the EU protection laws. If not, then if we're not showing that we can protect the data along the EU lines, then we could receive a fine up to 20 million euros, which is more than the grant that we received. And then it's important to know that this isn't my work. This is a large team, and we all work together. And Frank Verhulst, who's has just recently retired. And there's forced retirement in the Netherlands when you reach 65, but it's going up to 67. Henning Thiemeyer, Hennen El Maroon. You see Ryan Metzel on the list. Ryan has helped tremendously in terms of data collection. And then we have collaborators, and Manon Hilligers is the new chair of the department as of this year, who's really good. And then our funding, which is always a challenge, as you know, here. Thank you very much.
Thank you, Tanya. That was fabulous, really exciting. I'd like to open it up for questions, and I can pass around the microphone.
Thank you for the talk. A very specific question. With the polygenic risk and the lack of finding in the schizophrenia population on the white matter, did you also look at gray matter and total volume, or because?
Yeah, we also looked at the structure, and we had essentially the same findings. So I would have predicted the most consistent finding in schizophrenia is enlarged ventricles. So I would have expected that of anything to be different. We didn't find any difference related to the polygenic risk score in structure or DTI.
This was at what age?
This was at 9 to 11 years of age. So there's a number of reasons why that might be. I mean, it's before the onset, and you're just a polygenic risk, and most of them are not going to develop schizophrenia. So it could be related to actually trait rather than state.
Along those lines, the group that you're defining as the schizophrenia polygenic, is that defined on parameters at 5, 6 years of age and the same parameters throughout, or are they based on the oldest time that you've assessed and then carried backwards? Because things are going to get more specific as the kids get older.
So
what exactly are you
so
seeing?
the independent sample uses from the PGC, which are people who have a diagnosis, adults mainly. Maybe there's some adolescents or whatever, but they're adults diagnosed with schizophrenia. So it's the schizophrenia controls that through the Psychiatric Genomics Consortium, they've pooled all these individuals. And that's used to calculate the weighting for each SNP that is then applied to the children in Generation R. And the blood, was cord blood, but it should be the same. So the epigenetics that people are getting into would be a different question related to age.
Are you seeing any difference between the European population versus the immigrants in terms of those schizophrenia scores, given that the immigrants are at higher risk and may have a different symptom mix?
It's a good question. We have the data, but we haven't looked at that on immigrant
Lots of things that haven't been looked
data.
at yet.
Yeah. We being in the Netherlands, as people know what's going on in Syria, we do have a large influx of refugees from both from war-torn countries like Syria and Iraq, but also refugees, what do you call it, economic refugees, whatever, who are coming from different countries because their situation is very poor. So, but yeah, we're seeing that, and especially in Rotterdam, since it's very ethnically diverse, so people tend to come there. But those individuals will be much younger than, because Generation R has its cohort, and we're moving forward. But there's a new cohort starting called Generation R Next, where they want to do where they're doing more in-depth measures, which some of those refugees are going to fall into that group.
So your graphs go out to 18, the things that you've plotted. Do you have funding to continue the study into the next wave?
If we're allowed to do data sharing, and we don't have to give the money back to NIDA, then we'll have imaging funding for the coming 5 years, and a data manager, which is, as you know, extremely important, because a lot of the data like XNet server and everything, Ryan's doing, and we're working with Radiology's IT department on our XNet server. But the next wave of the study, which we're going to be meeting about, the Dutch government has funded consistently each wave, so I have no reason to believe that they'll stop funding it. It's been very productive and a good study, but you never know. Things are, so we do it in waves. So right now we're on the 13 to 14 wave. We'll probably go to 15. That's funded. The next wave will be the older adolescents, that's really an exciting wave, and we really would love to have funding at that point to do on everyone measures of psychopathology. and unclear which, but it would be nice to do, we were talking about the kitty SADs, but it would be nice to have some type of metric that gives a much better population-based measures of psychopathology. So we're looking into that. Ben Leahy and University of Chicago has some ideas of what he thinks we should use, and we're kind of going to be exploring that as we start planning that wave.
And a different question, if I may. You talked about diagnoses. Do you have permission to access their Dutch medical records?
We do. So all medical records in the Netherlands are via the huisarts, the family practice physician. And for our autism diagnosis, we received those through, we did screening from those who were high on the SRS and other measures, and then we did a random sample of just random, and we collected medical records from the physicians because we started to do ADI and ADOS on those children, and we had many families say, I already did that, I don't want to do it again. So then we realized that we should just go to the warehouser of all medical data, which is the family physician. And I think as the children go on, we would like to do much more of that and collect, and actually not just for mental health, but also for physical health and other parameters. That data is very valuable.
So this would include people that you've lost contact with who are still in the Dutch healthcare system?
That's a good question. So we have, I imagine if they, I don't know. Actually, I don't know
and the
about that.
Yeah, the final question would be, is there an explicit mechanism for people saying, I want to withdraw from the study?
Yes, and that's actually one of the things that that we have to work through with the NIH, because the NIH is in their data submission agreement, if a participant withdraws from the study, they in the NDA, what was NDAR but now NDA, a National Data Archive, they just remove it, but they don't inform everyone. They say we just remove it, but under Dutch law or EU law, not only do they have to remove it, but they have to request that all other researchers remove it. If there's a study that's published or about ready to be published, and then that can stay. Obviously, you can't remove it. But if they're currently working on data, we have to ask those research sites to remove that data. So those are things that there's laws in the Netherlands that are more, in the EU that are more strict.
Can they opt back in? Or can you have permission to contact them in the future to see if they're
we can send them a letter at a future wave to see if they're interested.
Do you have an estimate on the number of families that have withdrawn? I mean, you've lost about 30% of your cohort at the start, but I assume it's a small, like a handful of people? It's
a small handful of people who have done that. I don't know the exact numbers, but usually we can't, they don't answer the invitation letter or can't get a hold of them, or they've moved. And even though, the Netherlands has a very good system of registering people. So if I were to move to a different place in Rotterdam, I would have to go to the Stadswinkel, which is the city store, I guess would be a direct translation, but it's the courthouse, and I would have to register within 5 days. So they keep track of everything, which is really helpful for population-based studies, and not very good if you're in Nazi Germany.
Thank you for just such an impressive body of work, which is going to be yielding fruit for years and years and years to come. And I think also, fruit which will directly affect how we think about what we need to do, clinically with patients and populations. being someone who's interested in cognition, I was particularly curious about the findings related to your highly behaviorally dysregulated group of kids, and I wanted to make sure I sort of understood them. At what age were you looking at the cognitive decline, the relative cognitive decline? At what age were those measures taken?
So it's a very good point. The behavior and the behavior measures of the CBCL and the first non-verbal IQ measure, in which was 11 points lower, was done at age 6. Right around the same time. They're sent the CBCL by mail. The neuropsychological assessment, which was done actually in a subgroup of people, of the children, because that was in our pilot study, that was associated with the imaging study, that was done starting at 6, but going all the way up to age 9. So that was on average 2 years later, that neuropsychological. We are currently collecting WISC-5 data on all children within the cohort. And so one of the questions will be, we know that, we already know that the stability of this dysregulation score isn't very good at 1.5 when our first CBCL measure was done. But also for aggression, we find that stabilizes more around 6 to 9. So it'll be interesting. It might be that it's just those symptoms were just very unstable and just developmental noise or of some kind.
So you're looking to see now if you can sort of replicate in an age group and where you think this behavioral dysregulation measure has now become more reliable and more stable, and to see what the relationship with cognition will be at that point in time.
Right. We want to look at trajectories.
Yeah.
So very possibly within this subgroup of was only 2% of the children, a subgroup of them will likely have problems, but maybe there's a subgroup. Yeah, actually, I'm curious what's going to come out. I want to know what the reason is why we didn't see the findings that I would have expected to be there. What happens to these children?
On the imaging data.
On imaging data, but also on behavior and cognition.
Yeah.
So you could see that in a subgroup of those children, because not everyone participated in imaging, so there could be some bias there in that sample. The neuropsychological assessment was different than I expected it to be still across the board lower, but it's not.
Right.
So it'll be interesting to see in the larger cohort, when we include everyone except those who have dropped out, which you have a child with severe behavior problems, you're more likely to drop out. What happens to their behavior and cognition as they go into 13 and 14, which we're collecting now, along with the WISC-5. So I had 4 different hypotheses of things that could happen. One is that it's just not stable. The behavior is not stable, and so we don't find any findings because the behavior is not stable. The second is that there's just heterogeneity in the group because they go all different directions, all different disorders. The third is that there's developmental noise. So you can think of a growth chart. If you're measuring something in the growth spurt, children vary in when they have, the growth chart looks the same, but some children may start earlier, some start later, and so this might be developmental differences. And the fourth is based on a recent study also by with Ryan Metzel is the first, and I'm the last, where American Journal, where we show that behavior, I don't want to say behavior changes the brain, but it was a cross-lag model, but it wasn't brain predicting later behavior, it was behavior predicting later brain differences, but behavior is in the brain. So I say we couldn't measure it on an earlier measure for some reason. It does show up at 9. So that could be because of environmental influences. It could be because of some type of behavioral kindling. where if you do a behavior a lot, then it just becomes more ingrained, and then you see this activity-dependent changes in the brain by doing the behavior. So then the question is, which is why it's fun to see Katie do so many intervention studies, is if you intervene, does it then change brain? Which I think would be a fascinating thing to see.
I agree. I think this current wave is going to be really exciting. You haven't really looked at the 13, 14, 15-year-olds yet, but that's
we're
kind of
still
like
collecting the data.
The time, right, of the onset of our, like, all the disorders most of us are interested in, and you'll be able to look back. So now there's this developmental timing, and why do certain disorders emerge at that time? And I just feel like some of these questions are going to be explained by your study that we've been wondering about for years.
Explained, I like that word. It's very optimistic.
She's going to figure it all out for us. Other questions?
I think people are hungry.
We're pretty impressed. Thank you so much, Tanya, for being here, and this was really fabulous. Let's give her another hand.
You have been disconnected from the meeting.